non-desired effects. Uh, Anyone has any questions so far?
Sir, Ruchira here. Previously, I forgot to introduce. Sir, I want to ask one thing. Sir,
Sure.
before inventing a drug, some drug development process or testing procedures are done. So why in thalidomide it was not done? So much disadvantages and
No, yeah, a fantastic question actually. Okay, so this is where I think now you guys are very critical. Okay, so when you're doing clinical trials, say first in human studies, okay, so you decide a population you want to test your small molecule on, right? So what do you do? Usually you take healthy volunteers, right, for phase one trials, etc.
yeah,
So in phase one trials, again, look at the probability, right? You won't be using lot of, you'll be going for healthy volunteers and you won't be testing it on, say, pregnant women, okay? So what typically would have happened, again, I don't know exactly what happened with thalidomide. What would have happened is, say I consumed the drug and I got the anti-nausea effect with thalidomide, right? I extrapolated that, okay, it's helping me, it would help you, it should help pregnant women as well. So the patient population was extrapolated. They were never tested. First case, okay? And if you see, the defects were recognized only after childbirth, right? So, and clinical trials at that stage, it is 1962, right? They weren't thorough to begin with, and they don't last that long. That's why these days you often would have heard of phase four trials, right? It means like after phase three, after approval, right? They keep noticing side effects. And if you have severe side effects, they withdraw the drug. Right. So this is where I think FDA has gotten stricter with time because each of these learnings, however horrific they are learnings, right? So you try to learn from these instances and we have gotten better. But that being said, not all the drugs that get FDA approval are foolproof. They might have some other off target, which is long-term effect, which we haven't noticed because we don't do clinical trials for that long. And also the population we choose may not be appropriate. You might have an off target effect in a population which was never part of your clinical studies, right? So did that answer your question?
yeah, sir.
Cool. Yeah. Yeah. Any other question you're welcome to ask because it might just get a bit more heavy, I would say. Okay.
I'm Dr. Mala Shankar.
Yes.
I just want to add to your thing or whatever you have said, you have told is perfect. But this one, the thalidomide tragedy, after the children were born with limb defects, they went back and it is like a postmortem.
Yes.
So
Yes. Yes.
after the children were born with so much of limb abnormalities,
Sure.
then they went back and they found that these people had taken thalidomide for their morning sickness.
Exactly. So it was withdrawn in 1962, as I said. So that's a problem, as you rightly kind of post-mortem because kids, many of them died actually. But yeah, this is a very sad incident, horrific incident, right? Because yeah, I hope this never repeats, but there is learning from each step. So that's why I think even FDA has gotten stricter now. And even with these glues going into clinical trials, that's why there's so much focus on safety now. Because these have history, right? They can degrade anything non-specific and they can have severe teratogenic effects. That's why they're being screened very strictly now, right? Yeah, thank you, Dr. Malaya. Okay. So now I'll move on to PROTACs. So PROTAC stands for proteolysis targeting chimera. So these are heterobiunit molecules. So I've just copied this flowchart from one of the papers listed here. So this shows you a nice flow of evolution of PROTACs. So the first PROTAC technology came from Craig Cruz and Deschares' lab, and they showed that if you have a peptide which bound to a CF, which is an E3 ligase, so beta TRC is kind of an E3 ligase, and then you try to engage MET-AP2, you can selectively degrade it. As I said, you need two arms, like two ligands, which when linked by a linker, you can bring the two proteins together and artificially mediate its ubiquitination and degradation. So this was the first time the term PROTAC was coined. And the first PROTAC that induces the degradation of AR, which is androgen receptor and ER, but also like from the same lab, but it came in 2003. So I think for the PROTAC technologies, the Cruise lab has been a pioneer. So So they have come up with several innovations. And later they started using more and more E3s. So this time they started using FKBP12, a different E3 ligase, and also by recruiting another E3 ligase called as VHL. So you can see that the technology can be expanded to E3s, many E3s, and you can still have the desired effect, right? That's the degradation of the target protein. The first small molecule protein, because here these were all peptide based. The first small molecule protein came in 2008, and it was meant for degradation of MDM2. And then, as I said, in 2010, the thalidomide mode of action was recognized that it binds to cereblon. So now, because you have one arm thalidomide, they started using thalidomide as the cereblon recruiter, and they found other arms for whatever protein you want to degrade and started making PROTACs together. They also made different versions. So different versions of PROTACs are called by different names because each lab, when they come up with a PROTAC, they don't want to use PROTACs. So they come up with cool names like, for example, Sniper. So these actually employ E3 ligases called as IAPs. So the same concept, but different E3s, right? And here, first molecule using E3 ligase VHL. So in 2013, a first company was again brought up by Craig Crew's lab that actually tried to make drugs using PROTAC technology. So this is a very big step for our field. And in 2014, it was recognized as like lenalidomide was shown to be a molecular glue, and that went into clinical trials. And as you see more and more cereblon-based, thalidomide-based ligands now starting to function as the cereblon recruiter and PROTACs. Although thalidomide on its own is a molecular glue, but if you tag that molecular glue with a linker and the POI you want to, like a binder of a POI, now it becomes a PROTAC. So thalidomide is serving as a E3 handle. So it's called as E3 handle because it's going to engage E3. That's the cereblon in this case. The first PROTAC that is came into clinical trial is this ARV 110 and 471. They are for the AR and ER indications. Like you want to degrade androgen receptor and ER. Again, from Arvinas. And now there's so many other PROTACs in clinical trials. And until now, there's nothing that has cleared the clinical trials, and many are in phase 2 and phase 3. We hope that something comes through because it's always the first one that struggles the most. Once the first one is approved, the floodgates open because there's so much learning in the process of clinical trials as well. We can then apply it very fast on others. So hopefully very soon we'll have PROTACs coming through. That's why I said you clinicians will have to come across these small molecular glues and PROTACs a lot in the future, hopefully. Okay, so initially I told you PROTACs work by forming ternary complex. This is true even for the molecular glues. But in this picture, I'm showing you a PROTAC. So in this, if you look at this VH032, so this is the E3 handle that engages the E3 ligase called VHL. And this is the JQ1 moiety or the ligand that engages BRD. Okay. So, and here, this linker in the middle, right? So this is the first structure that came in 2017. And this is a ternary structure again, because you have the BRD4, which is the protein of interest. You have the PROTAC and you have the E3 ligase. So this is the VHL, and these are the adapter proteins which are necessary to purify this protein. That's why it's shown as a complex. If you could purify just VHL on its own, you could have just had this structure, but it's because you cannot recombinantly purify VHL alone. It's purified as a complex. And that's why in the structure, you see all of them. Okay. And this is a crystal structure. And that's why you see this density, nice density from the crystal structure. And this molecule is called as MZ1, right? And if you look into the PP, okay, when I say use the term PPI a lot, it means protein-protein interaction. What you would think is when they recruit to each other, you will think the BRD4 is separate, the VHL is separate, the linker will be hanging in the middle. Will be close enough so that ubiquitination happens. But in fact, it was seen that when these come together, they start interacting with each other, VHL and BRD in this case. That's why you see this nice interactions, right, between VHL and the BRD. And you can see even here interactions. So the linker is actually forming a scaffold on which these interactions are building up. And these interactions are very critical in maintaining the ternary complex long enough for the ubiquitination to happen, right? Now like several terms come up. So if you think of a PROTAC that can bind to protein A and C, so you can have two events. First, it can either bind to A or it can bind to C. And then it will bind to A to form this ternary complex. So irrespective of which path you follow, the end product is the same complex, right? And you can say, like, if you were to coin a term to define how strong the complex is, whether you form more interactions or less interactions, that's where this term cooperativity alpha comes. Okay. So the alpha is defined as the ratio of the dissociation constant of binary to the dissociation constant of ternary. Again, I'm assuming you're all beginners, so I'll give you why it's the so dissociation constant means it will give you an idea of the affinity, how soon it dissociates. Okay, so when I say it's a strong binder, it will have a very small KD. Say, for example, 1 micromolar KD is weaker than 1 nanomolar KD. Okay, so this is the number is opposite to the strength. Okay, so what this essentially means is that the ternary complex is stronger in terms of affinity than binary complex. Okay. And this is the term. And if the alpha is over 1, it's called positively cooperative. It means it's forming better interactions. Whereas if alpha is less than 1, it's negatively cooperative. It means it doesn't form a very stable complex. To give you more idea of how it would look like, say again, if the linker is not bringing the protein strong, like close enough, you won't form very good PPIs, right? So then you would have a negative or even zero cooperativity, right? So it means that they're just coming together, but they'll fall apart very soon. So what happens when they fall apart soon is you don't give the E3 enough time to add the ubiquitin onto the target, right? Whereas in this case, you see that the PROTAC brings the two together and helps it form very good protein-protein interaction. And this interaction is keeping it in that ternary state for longer. And that leads to effective ubiquitination and selective degradation as well. Yeah, so I think if anyone has a doubt until now, I can wait and take them before I go to the next slide.
Sir,
Yes?
this is known as docking, right?
Okay, docking is a process you use in simulations. Okay, so I think if you have a structure of a small molecule, then you can use, if you have a structure, crystal structure, then you can predict how another small molecule will bind the same pocket. So that is known as docking, but this is not a docking. This is a visualization of how actually two proteins come together. Did that answer your question?
Yes, sir, I got it. Thank you, sir.
Sure, no worries. I realize I have 15 more minutes. There's plenty to go with the rest of the slides. So I've tried to keep this as brief as possible so I don't want to load you with too much information. My intention is to give you an introduction into this technology so that when you come across these in the future, you will have an idea about these. Okay, so if you have no other question, okay, so what is the consequence of cooperativity? Okay, so this figure in the left, right? So you might have the same key, like you have different keys as you see here, okay? But based on which E3 you're using, so you have several E3s as I said, right? Only when you have the right E3, you might form the right. So although you're using the same key, all of them will fit on their own. But the problem is only the right pair of E3 and target will give you the optimal degradation because they together can form the complex longer and hence you can have degradation. That's why there's so much effort to explore several E3s, right? So why not stick with one E3 to degrade all the proteins? So that's a common question. So this is where the selectivity comes in, right? And assume, say you have the same lever and now you're changing different E3s. That is again, you will get the selectivity. So this is called an E3 layered selectivity because although this key, so assume I have 5 targets, okay? All of them use the same lever, but I have different E3s. Not all the 5 targets will be degraded because all of them do not form the same PPI. Only the select E3s along with the target which form the right PPI will be subjected to ubiquitination and degradation. An example of that is shown in this paper, okay? So palbociclib is the lever that binds to all the kinases. There's several kinases that bind to this ligand, palbociclib, okay? But interestingly, when they use different E3s, in this case, they screen for cereblon, IAP, MDM2, and VHL, right? I told you, right? There's several kinases. So each point here is uh, like either CDK6 or CDK4. They're only monitoring two, but they have different potency in terms of degradation and selectivity as well. For example, this one is degrading both CDK4, like only the VHL PROTAC is degrading CDK4, right? And CDK6 effectively. Whereas this one, or maybe let us take this molecule, right? So this is a VHL-based PROTAC, which is selectively degrading CDK6, but it is not degrading CDK4, as you see from the axis. So the axis represents what percentage of the protein is degraded. Higher the percentage, higher the potency, right? So this molecule here is selective towards CDK6 than CDK4. So you see that VHL1 is very selective, whereas the cereblon one is not so much. So they have tried different combinations of the degraders, and they show that you can achieve this E3 layered selectivity, which can actually differentiate targets even if you have a promiscuous binder, right? And this has huge implications because say if you have kinase binder, say palbociclib. So actually this is also in clinical trials now. So this is too specific. Say if you have ATP analog, right? So ATP is a favorite analog of inhibitors, right? That you use for kinases, right? But the problem is you will degrade if you're using as a, yeah, sorry, I don't know if there's a question or is it just noise? Yeah. Okay. So if you have a small molecule that can bind to all the kinases, say small molecule ATP analog, which can bind to all the ligase, sorry, all the kinases, and if you make a PROTAC out of it, you don't want all the kinases to be inhibited or degraded. The inhibitors can't give you that control, whereas the degraders can give you that control based on the E3 you choose, right? So this is where you get an additional advantage from this technology over inhibitors. Yeah, so if we look into the literature, right, like already PROTACs have been used a lot to degrade all these targets. So in the x-axis, you have the targets, right? And there are several PROTACs made for those. But interestingly, to degrade all these targets, mostly it's been cereblon or VHL, and to some extent IAP. And there has been a great effort to explore new and new E3s, that being said. Okay. So as I said, the question is, why do we need more E3s to degrade all these, right? Now, one of the questions, sorry, I forgot the first question who I got from. They asked the organ selectivity, right? So assume you have an E3 ligase only in heart, okay? And you want to degrade a particular kinase only in heart. Then just choose that E3 ligase. You'll degrade only the protein in that heart, not other organs, right? So this is where tissue selectivity comes in, right? So you get a choice based on your choice of E3, you can modulate the function of your PROTAC. The target selectivity already answered, right? So the cooperativity defines how, like even if you're using a promiscuous binder, you can get an E3 layered selectivity. Disease selectivity is also interesting because this is a classic example of, say, cancer, right? So cancer cells might be overexpressing a particular kind of E3, which a normal cell is not. So if you're degrading a protein of interest, if you are using a PROTAC, it will selectively, or a molecular degrader, so it will only degrade in that cancer cell and not harm normal cells. This is where you can get away with lower toxicity, right? So these are all the potential advantages that these degraders can offer. Yeah. Okay, so with respect to how a degrader is made, right? So there's several steps and different departments in an R&D. So I work in drug discovery. So we do all these things actually. And we try to make a lead molecule. So we do the hit to lead optimization, and then the leads go for clinical trials. So we just hand over the leads over to the clinical trials. Okay. So the PROTAC, in this example, I've shown PROTAC. So but all degraders typically go through the same. So what are the desired features of a degrader? You want it to be soluble and stable so that it can pass the cell. So then you need the permeability as well. So this is where you need to do different assays to check for permeability. You have other parameters like efflux ratio. I won't be talking too much about this, but you need to take care of several problems that might be associated with these. Then you need to check binary engagement. What does binary means? As I said, you need to engage, like PROTAC has two arms, right? You need to be sure that once inside cells, they're binding the two proteins individually. So that's the binary engagement. So you have different assays to monitor that, right? And then you will see if once they form the binary, can they form ternary as well? Then you assess the strength of the, like there are several assays to do that, and then you assess the strength of the complex using, say, cooperativity or intensity, different parameters. And then you check, okay, they're forming complex, but are they getting ubiquitinated? You need to check that as well, because not all the complexes form ubiquitination product as well. So there's several hiccups, you could say, or hurdles that you need to cross to get to the desired target degradation that you can also monitor in cell, right? So it's quite complex in the sense getting molecule is one, you need to also monitor the mechanism of action. Like even for inhibitors, it's not so straightforward. The fortunate thing is we've learned a lot from inhibitors and started incorporating those steps in here as well, right? So physicochemical characteristics, etc. So this is the general flow of how a PROTAC design would work, right?
Excuse me, sir.
Yeah.
Sir, due to maximum chemicals are used in the process, so degradation might be fast.
Sorry, I didn't follow. Can you repeat the question, please?
So there are many chemicals that are used in the development involves multiple blocks. So, so the chemicals which are used, so their maybe degradation may be very fast depending upon the process. So if it
Okay,
affects
so
the,
so the degradation would, yeah. So that okay. So if you, okay, maybe let me paraphrase this, and you can answer me if that's the question. So you want to ask if you're using a higher concentration, would you degrade better? Is that the question?
yes, sir. And it
Okay.
affects the molecules also, I think. So, so if the final result is pure form or it is affected form.
So we are talking about degradation of the protein of interest, not the small molecule. Okay. So if you're using, so one of the bottlenecks is using very high concentration of any degrader is first solubility. You might not be able to get that much inside the cell or the desired tissue. This is where the term bioavailability comes in. Like you might be taking paracetamol, right? But not all of it gets into the systemic circulation or the blood, right? Only a fraction of it gets in, but that's good enough to give you the activity. The same liabilities are around the degraders as well. So there's a lot of in vivo work that goes in to determine what fraction of your uh, inhibitor or degrader can get into the tissue of interest, right? So that's one challenge. The other thing is, even if it gets into the tissue, you need to be sure it's forming very effective ternary complex, it's ubiquitinating the target and degrading the target. Otherwise, it's of no use, right? So this is where you do several in vitro assays and then in vivo assays to make sure you are seeing the desired effect, but at as low concentration as possible. You need to keep the eventual goal is to take as little as possible so that first you don't have off targets, because when you take a lot of that drug, you might be hitting other proteins as well, right? So that's not the desired effect. So then you get toxicity, right? So the intention is try to minimize the dose all along. How do you do that? By making your compound as potent as possible. Did that answer?
Yes, sir.
Okay, cool. So these are just some examples of degraders in clinic. So on the left, we have molecular glue examples. As I said, most of them are in phase 1 and phase 2. And again, this paper is slightly older, 2023. Since then, one molecule at least has gone to phase 3. Uh, Again, these are the PROTACs. Arvinas is the pioneer for the PROTAC technology. And again, all of these are in clinical trials. We're still waiting for something that, okay, for example, the lenalidomide I said in the beginning, right? That's already a drug, but you wouldn't, but it's not a PROTAC. That's again, serendipitous, whatever it is. It was being used for a long time and it's already in clinic for cancer treatment. But all these new models, new PROTACs, new glues that have been designed, several of them are in clinic. So I wouldn't say a lot of them have clear that hurdle. Once they do, you will see a flurry of molecules coming as drugs to you. So very exciting time indeed. Okay,
Excuse
so
me, sir.
now, yes,
Sir, I have one doubt that before conducting the clinical trials, we will also consider the dosage form, no, right?
exactly. Yeah,
So we will text all the like parentals, orals, like
yes,
sublingual, all
yes,
the drugs
yes,
we will, yes,
yes,
all the
exactly.
drugs
All
we
will
will
be
text.
tested. Exactly,
Okay.
all will be tested.
Yes,
So
sir.
all these drugs you have, right? Not all of them are oral. So it all depends actually. So sometimes you get away with things that, say if you're taking a drug and you don't have to take another dose for 10 days, you're okay with injection, right? So that intravenous route is fine for that. But if you are taking something that after 6 hours you need to take again, you can't keep having injection, right? So that's where I feel the dose, the route of administration is dependent on the mechanism of action as well. So it all depends what target you're hitting, what you want to do. So the dose, all of these are optimized in the in vivo studies. Yeah. Okay. So I see I have last 3 minutes. So I'll quickly run through the two things. So today I talked about big like protacs and glues that uses the ubiquitin proteasome system to degrade your target. But there's several new technologies that have come up which use one or the other pathway, not the ubiquitin proteasome system, like say lysosome targeting or antibody based or glue tag. So there's several other technologies that have come up that all try to degrade the protein of interest, right? So they're all degraders, but they don't use the ubiquitin proteasome system. They have different ways, phagosomes or yeah, as you see here, several others. And if you can degrade, you can do more things as well. So you can do other PTMs as well, post-translational modification. Ubiquitin is a kind of a PTM that leads to degradation, but you have other post-translation effects as well. Say you can phosphorylate, and what can be the desired effect? So once you form a new PPI, they can change the stability of protein, or they can change the localization of protein. You can go from cytoplasm into the nucleus, or you can have an altered activity as well. Maybe now it's either hyperactive, or instead of doing A, it can do B as well. So there's several things you can change using this other modality. They're all post-translation modifications. So examples being, so of course, I talked about this PROTAC, molecular glue, etc. So these are all degraders. You can also have kinases which can phosphorylate, they're all proximity inducing modalities, right? So this is just a list of all the other PTMs you can do, right? So with this, I would just like to summarize. I gave you an overview of how these degraders work, how it is overcoming certain challenges that the small molecule inhibitors have, right? And yeah, I think pretty much other things have covered. So as I said, several of them on clinical trials
Sir,
and
excuse me, one last question.
yeah,
Sir,
sure, sure.
AI is very important in this. Is the AI
Sorry, again, it's a bit cracking. Can you repeat the question, please? Hello.
hello?
Yeah.
Sir, is there an artificial intelligence is playing role in this AI? Is a part of this?
So AI is used in everything, okay, but I think the movies have exaggerated it, or again, it all depends. So it's not AI is everywhere, okay. So when I say that you want to make a small molecule which is specifically binding to one protein, AI is used there as well. Like there's a team of computational chemists here, so who use AI ML language to actually design. So you are just automating. So earlier, what used to happen is we would manually look at the structure and visualize, okay, if I add one functional group over here, it will enhance the binding. Now AI has taken over that intuition, right? So it's doing all of that. So you just give the two things together, it will suggest all the changes through AI. So AI is embedded in all. It has been there for a long time, but the programs have become more efficient. That's why it's becoming more and more popular, right? But the programs have been there since beginning, but you're now trying to automate as much as possible. So as to make it more efficient. So one person can now design 10 drugs, right? Rather earlier 10 people are required to design one drug. So I would like to like take one fear out in terms of like AI replacing you all. It will make you all efficient, I would say, right? So try to keep up with the technology. But that being said, you should know the software or the application that helps you do that. If you are averse to AI, you won't learn that, then you'll be in trouble. Right? Cool.
Yeah.
Yeah, I think that's the end of my talk. And yeah, I can spare a couple, like 5-10 minutes more. If you have any other questions, yeah, I welcome all. Yeah.
Sir, I just have a overall doubt. Like, is the clinical trials being conducted on the pregnant women and all?
It depends on what the indication is. Say if you're trying to treat something in a patient, right, which is who is a pregnant woman. So phase one is not on pregnant women. Okay, phase one always happens on healthy volunteers. It's from phase two you go to the right patient population. Of course, you will do it on pregnant women also, right? So that is if you're trying to address only that population. It is conducted depending on which drug you want to treat. Yeah. I hope that answered your question, right?
Yes, sir. Yes, yes.
Cool.
Sir, hi, I'm Shweta, sir. I
Yes,
have
sir.
one question. What you told about Protex and glue, they're already always they have worked on only on ubiquitin activity. They have not worked on a lysosomal activity.
No, that's where the LITAC is coming. I was discussing more about ubiquitin
Maybe
proteasome system today, but I think only in like 3 or 4 years back, the LITAC came into existence, which is lysosome targeting degradation. And since then, more and more technology like AppTag, then you have autophagy targeting degradation. So there's several versions of it that has come up now. And like all of these are now, because you can imagine something that came up 15 years ago is in clinical trials now. So these things are still concepts. They will take much longer to come into clinical trials, right? Because you will start learning about pros and cons of technology. There's so much to learn, but exciting term in terms of these ideas coming up, right? So you have a lot to look forward to.
Okay, sir. Thanks, sir.
Yeah, sure. Yeah, any more questions? So, Priyanka, I don't know if I'm having issues with the internet.
No, sir, your voice is totally audible.
Okay,
Yeah.
okay, cool.
Yeah.
No, because my video has been turned off, so I don't know if my internet is working fine. Okay, thanks.
Fine. I mean, it was working
Sure.
fine.
Cool.
Not so far.
sir, what message you can give to the your young aspiring in the
Sorry, again, can you repeat it?
sir your one message to young aspiring students who are interested in the pharmaceutical industry for their career?
Again, as I said, my path may not be the same as yours, right? I took a different path in the sense I did my master's, PhD. So I've been an expert. I wouldn't say an expert. I've been trying to learn a lot about a very narrow field, okay? But one recommendation I would give is you need not be a master of all the trades, but I think it's not a bad time to be jack of all trades, right? So you should try to learn about all technologies or all the disciplines because more and more it's becoming multidisciplinary. So you should try to learn a little of all. You need not master it, but at least you should be able to connect the dots when it matters the most. So try to be open to learning. So try to attend, ask people more and more, like of different disciplines. Again, I assume most of you are looking into clinical research, so that's a narrow discipline on its own. That being said, keep an open ear to say the drug discovery developers as well, in the sense you will learn a lot from them, and the same goes vice versa. So we have a constant collaboration with the clinicians as well, so as to see what problems we want to address. So yeah, this interdisciplinary goes beyond. So it, like for example, you might be a cell biologist, but that being said, be open to talking to a physicist or even to a chemist and understanding how a drug comes together, because all these things are synergistic, right? So you keep learning from each other and try to find out the right problems and the answers will follow. Yeah, again, a
Sir,
very vague
thanks
answer.
for your view.
Yeah, sure. Cool. Any other question?
Yeah, Sarath sir, I guess students are done with the questions. Thank you so much for joining us today and for valuable insights. Our
No, my
students,
pleasure. Yeah.
yes, sir. I know our students have learned a lot from your experience, and your advice will have a lasting impact on their journey. So we hope to see you soon again, sir, in future. Your contributions have sparkled a lot of interest in our students that I can see. So thank you
No,
so much.
sure. Thanks a lot. Yeah.
Yeah,
Thank you so much.
Yeah.
Thank you so much, students, for joining us today. Have a very nice evening, all of you, and sir as well.
Thank you, ma'am.
Yeah.
Thank
Thank you,
you.
Thank
sir,
you.
for your
Thank you, ma'am.
precious
Thank you,
time.
sir. Thank you, ma'am.
Thank you, ma'am. Thank you, sir.
Yeah.
Thank you,
Thank
ma'am.
You
Thank you,
all
sir.
have a good day and hope of a lot of success for you all. Thank you.
Thank you, sir.
Same.
Thank you, sir.
So Priyanka, I can disconnect the call, right?
Yes, sir. You can disconnect the call.
Okay, perfect. Thanks a lot.
Yes, sir.